Hi everyone, I'm Farhana. I'm one of the TAs for this course, and I'll be presenting to you the case studies for COVID-19. So hopefully you had a chance to look at them and to look at how to answer some of these questions. So before we take a look at the case studies, let's briefly talk from the very start a bit about COVID-19 and its epidemiological impact. So COVID-19 is a disease that is caused by infection of the SARS-CoV-2 virus, which belongs to the coronavirus family. SARS stands for severe acute respiratory syndrome, and CoV stands for coronavirus. And coronaviruses belong to a class of encapsulated positive single-stranded RNA viruses that have a diverse spectrum of natural hosts, humans being one of them. Some of the symptoms of COVID-19, I think we're somewhat familiar with this as we've been asked these symptoms when entering restaurants at some point. So fever over 100.4 degrees, cough or sore throat, new shortness of breath, chills. Some people may experience new muscle pain and also new loss of taste or smell, as well as nausea, vomiting, diarrhea, and new headache or excessive fatigue, and congestion or runny nose. COVID-19 symptoms typically occur 2 to 14 days after exposure to the virus, and most commonly after 4 to 5 days. So most people will experience mild to moderate symptoms during this period. The first COVID-19 symptoms to develop is likely to be cold-like symptoms, such as fever, which is a temporary increase in body temperature. And this differs from influenza, where the first symptom is typically a cough. Some patients may also experience a loss of taste or smell, as well as a decrease in white blood cells. And about 20% of people will experience acute respiratory distress syndrome after 1 week to 10 days. And this is where the lungs fail to provide the body with sufficient oxygen. So according to the Center for Disease Control and Prevention, CDC, around 20 to 42% of people hospitalized with COVID-19 experience this second stage 2 condition. And in stage 3, often about 1 to 2 weeks after symptom onset, patients experience worsening shortness of breath and hypoxia, along with subclinical elevations in the indicators of organ damage in cardiac, kidney, and liver. And doctors may admit people to an intensive care unit in these severe cases. So this data is extracted from the World Health Organization. Globally, as of March 29, 2023, when I collected this data from WHO, there have been 761,402,282 confirmed cumulative cases of COVID-19, including about 7 million cumulative deaths reported to the World Health Organization. And this figure is COVID-19 cases reported by WHO region and global deaths by 28-day intervals as of 26th February 2023. So this is 28-day interval. So you have data from December 2019 to January 30th, 2023. And the World Health Organization organized countries into regions: America, Southeast Asia, Europe, Eastern Mediterranean, Africa, Western Pacific. And this dark black line is the deaths, tracking the deaths along, I guess, globally. And at the regional level, the number of newly reported 28-day cases decreased across all WHO regions, as you can see. And the number of newly reported 28-day deaths also decreased across the 5 regions. So we can see here that around December 2021 to February, there was an increase in the number of cases, as well as an increase in the number of deaths. And then we have a dip in these cases, but in December of 2023, we see an increase as well, but within the Western Pacific region. So there may be several reasons for the dip in cases and deaths that we're seeing right now. And one explanation is more of the population is vaccinated, which is protective against new infections. And also there has been changes in COVID-19 testing policies in many jurisdictions since December 2021, and so case counts may be underestimated. So here in Canada, so this is as of March 27, 2023, we see a similar trend where we see the highest peak around December 2021 for cases, with other periods having rises and falls in case numbers. And for number of deaths in Canada due to COVID-19 as of March 27th, 2023, we can see that January 2022 seems to be a highest peak for number of deaths, and the number of deaths have been around and under 400 all through 2022 and now 2023. So, what are the factors that are causing these peaks um, in cases and deaths from COVID-19? And how do we avoid a situation like what happened in December 2021? Um, so those are the questions that would be interesting to answer. Of note, the shaded area for figures 2A and 2B represents a period of accumulating data where it is known or expected that cases and severe outcomes have occurred but not have yet been reported nationally. So these data may not completely reflect what is happening all over Canada. And also the Canadian government website also indicated that due to changes in COVID-19 testing policies in many jurisdictions since December 2021, case counts are all maybe underestimated. So when we look at the number of cases, hospitalizations, and deaths by vaccination status, the Public Health Agency of Canada reported that between August 29, 2022 and September 25th, 2022, unvaccinated cases were 3 times more likely to be hospitalized and 5 times more likely to die from their illness compared to cases with a completed primary vaccine series, which means two vaccinations. So vaccinations seem to be a protective factor against more severe forms of viral infections. So this brings us to our first case study, which focuses on COVID-19 bivalent booster vaccines. So the case study, the patient here is experiencing, so I'll just read it first. So a 45-year-old woman approaches you in a community pharmacy who is unsure about receiving her bivalent COVID-19 vaccine. She's confused about whether she's eligible for her booster vaccine and which one she should get. She has heard so much information about COVID-19 variants and subvariants and asks you to clarify how the currently available vaccines are related to the virus variants. She received her last monovalent Pfizer BioNTech's COVID-19 booster 8 months ago. She has no chronic medical conditions and she doesn't understand how a booster vaccine works. She wants to know when and which vaccine she should receive. Your community pharmacy stocks Moderna's Spikevax, original and Omicron BA.1 COVID-19 vaccine, and original and Omicron BA.5 and .4, and Pfizer BioNTech's Comirnaty, original Omicron BA.1 and original Omicron BA.4 and .5. So in presenting this case study, briefly discuss mechanisms and effectiveness for bivalent mRNA vaccines, and then outline information that you would provide to this patient with discussions about the results of efficacy and safety of booster vaccines. So in summary, it seems that the patient here is experiencing some confusion regarding COVID-19 variants and subvariants and how currently available vaccines relate to the virus variants. She also lacks understanding of how booster vaccines work, particularly bivalent COVID-19 vaccines. She said she received a monovalent booster 8 months ago, and she now wants to know if she should get a booster and which one she should get. Those are very good questions. So let's first start with demystifying COVID-19 variants. So SARS-CoV-2 is an RNA virus that has been evolving since its discovery in December 2019. And as a virus mutates through the replication process, there may be mutations to the viral genome that result in a variant of the original virus. Mutations can be neutral, beneficial, or harmful to an organism depending on the specific mutation and where in the genome the nucleotide substitution, addition, or deletion occurs. RNA viruses are more likely than DNA viruses to develop variants since copying RNA is more prone to mistakes than copying DNA. So here is a schematic representation of the SARS-CoV-2 structure showing the exterior envelope, um, the membrane spike glycoprotein shown here, and the nucleocapsid and the RNA viral genome. And here we have the schematic representation of the SARS-CoV-2 genome organization, which is about 30 kilobase pairs. You can see that there's two open large reading frames, ORF1A and ORF1B, which encode the non-structural proteins separated by ribosome frameshift site, and there's genes encoding the structural proteins, this S for spike, E for envelope, M for membrane, and the N for nucleocapsid. And up here we have the schematic representation of the SARS-CoV-2 spike protein. And we can see that the spike protein has two subunits, S1 and S2, and we have this crystallographic structure of the SARS-CoV-2 spike protein. So a typical SARS-CoV-2 virus accumulates 1 to 2 nucleotide mutations in its genome per month, which is about half the rate of influenza and a quarter of the rate of HIV. SARS-CoV-2 spike protein is about 1,273 amino acids long and is the main target of the current COVID-19 mRNA vaccines. It is a portion of the virus that recognizes and binds to host cellular receptors, which is the angiotensin converting enzyme 2, ACE2 receptor, and mediates viral entry, as you can see here. It's binding to ACE2, and this is how it's mediating viral entry. SARS-CoV-2 is unable to infect host cells without this receptor. So ACE2 is abundantly present in humans in the epithelia of the lung and small intestine, which might provide possible routes of entry for SARS-CoV-2. And because of this, mutations in the spike gene, particularly those that affect portions of the protein that are critical for pathogenesis and normal function, such as within the receptor binding domain, or those that cause conformational changes to the S protein, are of the greatest interest. And if these changes are not recognized by first wave antibodies, these mutations may provide an avenue for the virus to escape immunity to the original SARS-CoV-2 strain. So the World Health Organization and the World Health Organization evaluates SARS-CoV-2 variants circulating in the population and makes recommendations about the classification of these variants. So these variants can be classified into categories such as variants of interest, variants of concern, variants of high consequence, and variants being monitored. So variants of concern are those variants that have increasing transmissibility compared to the ancestral strain. They have more severe disease, for example, increased hospitalizations or deaths. And there is significant reduction in neutralization by antibodies generated during previous infections or vaccinations. And there is reduced effectiveness of treatments or vaccines or diagnostic detection failures. Greek letters are assigned to variants of concern, and we have variants of concern designated as alpha, beta, gamma, delta, Omicron BA1, and its subvariant Omicron BA2. I'll go more in detail about these subcategories of variants in the next slide. So in comparison to the Wuhan 1 strain, all of the variants of concern have a higher transmission efficacy, have lower protection from previous infection, and lower vaccine efficacy. The pathogenicity is higher, except for Omicron strains, and more monoclonal antibodies showed reduced efficacy against the newer variants. So the Pango lineage system allows us to be more specific in classifying lineages and sublineages of variants, because we will come to a point when we run out of Greek letters to assign to variants of concern. So the Pango lineage system is hierarchical like a family tree. Lineages are evolutionarily descendants of a parent lineage. So example, COVID-19 lineage are named using an alphabetical prefix such as B or BA and numerical suffix such as 0.1 or 0.1, 0.1, 0.5. A lineage may be described as a sublineage when it is being discussed in relation to its parent lineage. So example, when a new sublineage is defined, the Pango system assigns an additional number to the name of its parent lineage. So BA.2.75 is a sublineage of BA.2. And as the virus continues to change, the Pango lineage names can become very long. So lineages with longer names may be given alphabetical aliases and numbering continues. So BA stands for B.1.529, thus B.2 is the same as B.1.529, but additionally dot 2. So So in November 2021, the World Health Organization first identified the Omicron variant of SARS-CoV-2 as a variant of concern. This graph here shows the percentage of COVID-19 cases by the most prevalent lineage and week. This is surveillance data in Ontario from February 27th, 2022 to February 25th, 2023. So what we see here is that within 1 year, there were several new Omicron variants that have been identified in patients with COVID-19. So earlier last year, Omicron sublineages BA2 and BA1 was a dominant subvariant. Then between July and December 2022, the BA5 subvariant of Omicron was a dominant subvariant causing COVID-19 in most countries and showed significant escape from antibody neutralization when compared with previous SARS-CoV-2 variants. And the start of 2023 over here brings a new subvariant of Omicron known as XBB.1.5. The Omicron XBB.1.5 subvariant is a sublineage of XBB subvariant, which is a recombinant of two BA2 sublineages with an additional spike receptor binding domain change. And this mutation in the spike protein increases infectivity due to increased binding affinity the human ACE2 receptor. So if we zoom into the beginning of 2023, this is data specifically from January 1st to March 5th, 2023 of this year. And it shows the percentage of COVID-19 cases in Canada identified through whole genome sequencing, presented by variant and by week of sample collection. So nationally, clinical sequencing shows that XBB.1.5 has displaced previously dominant variants of BA.5 lineage. This XBB.1.5 subvariant has been named the Kraken subvariant in the media. Kraken, as some of you may know, is the name of a legendary Northern European giant sea monster, which was the topic of an apocalyptic poem by Lord Alfred Tennyson, published in 1830. So now the Kraken is the dominant subvariant of COVID-19. So switching gears to talk more about the vaccine, mRNA vaccines contain genetic instructions for making the COVID-19 spike protein, like I mentioned earlier. And this protein is found on the surface of the virus that causes COVID-19 and it allows entry into the host. So when a person is given the vaccine, their cells will read the genetic instruction like a recipe and produce a spike protein. And after this protein piece is made, the cell breaks down the instructions and gets rid of them. And then the cell displays the protein pieces on its surface. And when we are later infected with the coronavirus infection, our immune system recognizes that the protein does not belong within the host and begins building an immune response and making antibodies against this spike protein. And the mRNA vaccines, I wanted to know that mRNA vaccines do not interact with or alter your DNA in any way. There might be some misconception about that, but mRNA vaccines do not interact with your DNA. The quantity and quality of the antibodies that the body produces actually naturally decrease with time. And a booster dose is meant to increase levels of immune response after these have naturally waned. So as per the World Health Organization, booster doses are administered to a vaccinated population that has completed a primary vaccination series, which is currently 1 or 2 doses of EUA COVID-19 vaccine, depending on the product. When with time, the immunity and clinical protection has fallen below a rate deemed sufficient in that population. The objective of booster dose is to restore vaccine effectiveness from the deemed no longer sufficient. So first, we have the first vaccine dose that we all get. and which is part of the primary series. So the first vaccine dose, and then um, about 28 days later, we get the second dose, and that is when we complete our primary series for the COVID-19 vaccines. And after 2 months or more, immune protection decreases, and that's when we can get the booster dose. And I'll talk more about the different kinds of boosters. In January and February 2022, Pfizer BioNTech produced a bivalent vaccine containing 25 micrograms of mRNA um, directed against the ancestral strain of SARS-CoV-2 and 25 micrograms directed against the Omicron BA.1 strain. So we see that instead of one RNA sequence, the vaccine contains two RNA sequences for two different variants of the spike protein. And the vaccinated person then produces both of these variants of the spike proteins, and the body produces antibodies to these spike variants. So when the vaccinated person is infected by either the ancestral strain or the Omicron variant, they will produce the strong antibody response to both, and the virus will be eradicated before the infection can get to a severe point and have an effect on the individual. So this paper published in the New England Journal of Medicine demonstrates the effectiveness and safety of bivalent vaccines compared to the original monovalent vaccine. So here we have, they had 819 participants who had received two-dose primary series of the mRNA 1273 vaccine plus a booster dose at least 3 months earlier, were given a second booster with either the bivalent mRNA 1273.214 vaccine containing 25 micrograms of each ancestral Hu1 and Omicron B.1.529 spike mRNAs, or the monovalent 50 microgram mRNA monovalent booster containing just the Wuhan Hu strain. The primary objective were to assess the safety and reactogenicity and immunogenicity of mRNA bivalent at 28 days, as shown by non-inferior or superior neutralizing antibody responses against Omicron and non-inferior responses against ancestral SARS-CoV-2 with the D614G mutation. In terms of their results for efficacy, here A shows the level of neutralizing antibody titers against the Wuhan 1 ancestral strain. The presence and level of antibodies against the Wuhan 1 ancestral strain was higher with the bivalent vaccine than the monovalent vaccine in those with no previous infections, and those also who had a previous infection, which indicates that there were no decrements in the ancestral SARS-CoV-2 antibody response. B shows the level of neutralizing antibody titers against the Omicron BA.1 variant. Here we also see that the mRNA 1273.214 bivalent vaccine elicited a superior neutralizing antibody response against Omicron as compared with the monovalent vaccine 28 days after the booster dose. In terms of any adverse reactions to the bivalent booster relative to the monovalent booster, the safety and reactogenicity were similar with the two booster vaccines. So Health Canada approved two Pfizer-BioNTech Comirnaty bivalent COVID-19 vaccines authorized for use as a booster dose. So they have authorized original and Omicron BA.1, and this is authorized for use in individuals 12 years of age and older, and also original and Omicron BA.4/5, which is authorized for use in individuals 5 years of age and older. And only the BA.4/5 formulation is available for distribution in Canada. Moderna, there are two Moderna Spikevax bivalent COVID-19 vaccines authorized for use as a booster dose, which is original and Omicron BA.1, which is authorized for use in individuals 6 years of age and older, and the original and Omicron BA.4/5 authorized for use in adults 18 years of age and older. So current recommendations for Ontarians is it is recommended that everyone Ages 5 and older get one mRNA COVID-19 booster, original or bivalent, on or after September 1st, 2022. So the conditions are it has to be 6 months after a COVID-19 vaccine dose or a COVID-19 infection, and 3 months, 84 days may be considered based on age, risk, and health status. A bivalent mRNA vaccine is preferred. And the mRNA COVID-19 vaccines provide good protection against hospitalization and severe illnesses within 3 to 6 months following vaccination. So, based on the current recommendations, going back to the case study, the patient is eligible to receive any of the available bivalent boosters since more than 6 months has passed since her last monovalent booster dose.